All right, so the apply functions in R are incredibly useful, incredibly efficient, and kind of takes a while to get used to. So let's go over some of the common uses of the apply function. The main apply functions, there's actually a whole set of them, but the main ones are apply and tapply. There is also lapply and sapply, which I've used occasionally, but these are the two that have come in extremely useful to me in doing genomics analysis. So the apply function, essentially the idea is exactly what it sounds like. You want to apply a specific function to the entire data set. So in this case, the first argument is an array or even the matrix or the data frame that you want to apply the function to. The second is whether you want to apply this function to every row by noting 1, or to every column, or to every value inside this data frame. And then the third argument is the function that you actually want to perform. There are additional options to this function. After that, all the other options are related to this function that you provided in the third argument. And we'll see how we can use this to our benefit in a bit. The tapply is related. Again, the idea is to apply a function, but in this case, we're going to apply it not based on rows and columns. It's going to be based on groupings. So remember we talked about factors last module of how we can use it to group together our data sets. So maybe we can assign groupings to our data sets and perform operations. So the input in this case is usually a vector of values, and the second argument is actually how you group them. The lapply is the same idea, but instead of, again, because it's a list, it just performs the function to every member of the list. Okay, so let's continue on this idea of calculating the average for every row. Okay, so in this case, instead of looping through it, we're going to actually use an apply function. Okay, so again, just to remind you, this is our sample data, and now to perform this mean function to every row, we simply use the apply function. This is the name of the function, and then we provide the argument. In this case, it's our matrix. Over here is 1, which means every row. So 1 equals row, and 2, which would mean column. So if you put a 2 there, it would calculate the average of all the columns. And this is the name of the function you want to operate, and that's it. Okay, so this is a very efficient way to do this. Now, why would you want to do this? Now, if you think about it, if you want to calculate the average of every row, there's no reason to wait for one to be finished before you start the other, right? Because the average of one row has no effect on the average of the other rows. So why not just calculate them all at the same time? And that's where efficiency works out, because we're going to be working with data sets that have 20,000, 30,000, 50,000 genes. There's no reason to go through every single gene to perform a test or do some sort of calculations. Why not just run them all at the same time, make it really efficient? Actually, there are a lot of commercial packages that are available that actually take advantage of build specialized apply functions that can be run or executed on high performance computing, so that not only can you run this in parallel, but it actually learns or is aware of all the different computers that are around it, and it will actually spread the workload as well. So that gets pretty impressive and very efficient way of doing this. So let's do this on a data frame as well, right? So again, let's take this moment to review some of the ideas we talked about. Let's convert a matrix into a data frame. So to do this, we're going to use one of our as.dot functions. So remember, these as.dot allows us to convert one type of one class of a object to another. So this was a matrix, and now I've converted it into a data frame, and I've saved it as a new variable. Okay, so now what this allows me to do is have columns of different types, right? But for now, we're just putting it in a format that we get more comfortable with, because when we're loading in the data, this is what we will be seeing. Now, once we have this, we can also provide column names. So we're adding headers. So what I'm essentially doing is I'm making this into a data frame that we use in transcriptome a lot, where every column is a different experiment. So this is my control 1 and control 2, and treatment 1 and treatment 2. And the rows in our data set are all the different genes. In this case, I'm making all the rows into different genes and the different columns as the different experiments. So once you're done, you should see a data frame that looks something like this. Now, just remember, we've already covered this, but you can use these names to extract values. For example, you could say, I want the treatment 1 value for gene 3, and you can use the names for this as well, okay? Now, once we have this data frame, let's see if we want to now calculate the average of the values, okay? So these are my control values. So these are my control experiments. These are my biological replicates, right? 2 samples that were executed that have a control environment. And these are 2 biological replicates that have treatment environments. So now what I want to do is actually calculate for this gene 1, what's the average control value and what's the average treatment value. Okay, to do this, we will use the tapply function. Okay, so before I do that, I need to create a factor so I can define how to group the values. Now here, the order matters. So here, I'm going to use control as a way of defining that the first 2 values are identical. Okay, that means they are from the same sample, the biological replicates, right? So they have the same name, so they'll be grouped together. And these is another name that's repeated. So these 3rd and 4th value will be grouped together. So the first 2 are going to be control values, and the last 2 are going to be treatment values. So now what we can do is we can actually do the tapply function, and we're going to take the values of the first row. So now here, again, I'm using this as.numeric because remember, in data frames, every column is the same data type, but every row does not have to be the same data type. So if I don't do this, R is going to complain and say a row is not necessarily all numbers. So what here I'm telling it to do is saying, take the first row, and I'm telling you that these are numbers. Okay, so these are numerical values, and then this is my factor that I've created here. Okay, and this is my function. So for this vector of values, the first 2 are going to be calculated, the average, and the other 2 are going to be calculated, the average, and you should get something that looks like this. Okay, so here we've calculated the average control value for gene 1 and the average treatment value for gene 1 as well. Okay, so this is great, but again, how do we do this for every single gene? We could do this in a loop, right? We've talked about loops, but why don't we apply the tapply to every row? Okay, so this is where it gets a little bit tricky, but bear with me and you can hopefully figure this out. So what we're going to do is we're going to say apply to this data frame, every row, the function tapply. Now for tapply has its own arguments, but the first argument for tapply is the actual values, but the apply function is giving me that already, right? So the apply function is already going to give me the values. That's where I'm going to get it from here. So what I really need to just provide is the EXP groups and the mean. And so that's what I will do. I'll say EXP groups and mean. So again, what I'm doing here is I'm doing a tapply function based on the groupings and applying the function mean. But I'm doing this and I'm applying this function to every row in my data frame. What was the result? You will get basically the headers are a little bit shifted, but for every gene you will get an average control and treatment value provided to you